Hello, everyone. Thank you for attending today's session. It is our first in a series of webinars by Triverity Laboratories. And today's session is on pharmaceutical specifications, what they are, why they matter, and how to build them. Wendi Young is our CEO, is hosting today, and she will go through the presentation. Just a couple of housekeeping things, and the session will be recorded and we will share the recording with you after the event so you can refer back to it. We will also share the presentation, and all participants will be muted during the presentation. I'm not sure if you were able to hear the robot whenever we started there. Any questions that you have during the presentation, please add them to the Q&A panel. And at the end, we will have about 10 to 12 minute Q&A session at the end, and all will be addressed at the end of the webinar. And with that, I will let Wendi begin and start the presentation. Thanks for joining.
Thank you, Alex. Thanks everybody for joining us today. We're starting our webinar series with pharmaceutical specifications. This presentation is going to be really a broad stroke in specifications. We're not going to get into details because honestly we could do, and we are going to do several webinars on things like impurities and other topics like dissolution and things like that. So what we're going to talk about today, first we're going to start with the applicable regulations. We're going to talk about why specifications matter, the specification types that are required by ICH and different regulatory bodies, required tests, whether they're universal or specified tests, setting acceptance criteria, method applicability, and then also talk a little bit about product life cycle. So to get started on the applicable regulations and guidances, in CFR 211-160, that's where the regulation that states that you need to have a specification exists. In 165, it talks about the drug products that are manufactured needing to meet those specification requirements, needing to conform to specification for release. There are a lot of guidances in the USP regarding specifications, specifically around different dosage forms. You can see there, I just put USP 1 through 5. There's other ones too. USP 1's for injections and implanted drug products. I spent a lot of time in my previous job on USP 3 for topicals. There's also USP 561 for articles of botanical origin that talks about drug products that are manufactured from plant material. And then the ICH, honestly, I could have listed every single ICH. They all have touch on specifications in some way. The ICH Q6A and 6B are the guideline for specifications specifically. But there's also a lot in the pharmaceutical development, product life cycle, and things like that. So why do specifications matter? Really, it's because the specifications, it's the quality commitment that you're willing to stand behind for your drug product. It's what you provide to the FDA. It ensures that your drug product is acceptable for its intended use, and really is a key part of what the regulators approved as your established conditions. And established conditions are the documents and the information that you provide to like the FDA or another regulatory body that is something that if you're going to change it, you're going to have to get approval to be able to change it. And really, specifications drive batch disposition decisions. It's what you look at to determine whether or not a batch can be released to the market. So what is a pharmaceutical specification? Specifications establish the set of criteria to which a drug substance or drug product should conform to be considered acceptable for its intended use. So really it is the tests or the criteria or the parameters, however you want to call it, that you're testing. Then the methodology, a reference to the methodology that you're using, whether that's compendial or an in-house method, you need to state very specifically, this is the method that I'm going to use to test this. And then the acceptance criteria associated with each one of those tests. The different specification types are raw materials, so the excipients that go into your finished product, container closure, the packaging, in-process tests or bulk testing, drug substance release, drug product release, shelf life or stability, and then critical utilities. And critical utilities would be things like Wi-Fi water or compressed gases that you would use in your process. For our presentation here, we're focusing really on the drug substance and drug product release, but we're going to touch on just a little bit of the raw material and critical excipients or critical utilities. So this is an example of specification. It's just something that I put together. I chat GTP'd a fake drug name for it, and it's my background again is topical, so it's a cream. But I'm a visual person, so I think it helps sometimes if you're not familiar with specification to see what one actually looks like. So in the first column, you see the characteristic. That's the test. What test are you doing? In the middle column, it is the methodology that's being used, in this case, visual for color and appearance, and then some in-house test method references. And then in the last column, the acceptance criteria that you would assign to that test. So a little bit on raw material, container closure, and critical utility specifications. Raw material specifications, unless you're making your raw material yourself, you're typically buying it from somebody. And you can set your specifications usually based on what the manufacturer specification is. So if you're, where that kind of falls apart is if you're buying something that also is used in non-pharmaceutical materials, you really need to make sure that the specification they have is a pharmaceutical specification. A lot of times there's a USP NF monograph for a raw material, and you can use that monograph to set your specifications, and it makes it pretty clear. For container closures, packaging, typically you're looking at things like dimensions, visual defects, or functionality for more complicated container closures. And then critical utilities, you want to look at your critical quality attributes or your critical process parameters to set those specifications. Sometimes there's also a compendial standard for those critical utilities like the Wi-Fi water. So the specifications really are part of a total control strategy. I kind of like to look at specifications being the QC side of quality. So you have your quality assurance side, which is the development work that you've done, your method validations, your batch records, your facility SOPs, all of that. And then you have your specifications come in, you test, and then you're showing the QC side of it is the check at the end. Did all of the quality assurance work that we did result in a product that is as we would expect? So the specifications really should confirm the quality and not create it. You need to have a specification that you're pretty confident you put enough controls in that you're going to meet that every single time. So when you're designing your specification, there's a few things. We're going to get into building the specification a little bit later, but you want to look at the specification type. Are you doing an in-process specification? Is it a drug product release specification? You want to look at the dosage form. There's a lot of different tests that are required based on dosage form. You need to look at all the data that you have. And then also you want to look at the regulations like ICH, FDA regulations, or sometimes USP general chapters. What belongs on the specifications? You really want to include tests and criteria that are useful for ensuring the safety and efficacy and for consistent quality. So you want to look at the critical quality attributes of your product, and that really is what should drive what goes on your specifications. Your specs shouldn't attempt to fully characterize your product. And a good rule of thumb for this is if failing that test wouldn't change whether or not you would release the product to market, then you really need to reconsider whether it really belongs on your specification or not. There's a lot of universal tests. If you look in ICH Q6A, which is the specifications guidance from ICH, there's universal tests on there. And these are the ones that are kind of for every single dosage form, and then also for drug substance and drug product. So the first thing is description. And description would be color and appearance, maybe organoleptic properties, something like that. And this is something that I feel like we could do a whole entire webinar on problems that I have had over the years with a color and appearance specification. So as an example, you know, when you're doing a color specification, sometimes we have, we see colorless, or sorry, we see clear to yellow, right? So clear is not a color, colorless is a color, and clear is an appearance criteria. So what a good thing to do when you're setting your description specifications is to draft it, take your product, put it in front of somebody else or 2 or 3 other people and say, would you agree that this meets that specification? And really take a critical eye to it and say, how would somebody else interpret this? Because we do see a lot of challenges sometimes in getting products to meet the specification. You know, what is tan? If you're really getting into colors, then maybe you need to use something that's more qualitative for colors than just a visual tan or yellow. The other thing is identification is another universal test, and you need 2 forms of identification on your specifications for your API or your drug substance. Typically, we'll see on the drug substance specification a retention time or spectral match, as well as an FTIR ID test. And that's pretty common in the USP monographs. We also see for a drug product, typically a retention time and spectral match from your assay. So if your assay is HPLC DAD, we would see a UV spectral match as well as a retention time match for that to satisfy that criteria. The assay test is just the assay test, typically 97 to 103% on drug substance and 90 to 110 on drug product on shelf life. And then impurity and degradants, and we're going to get into impurity and degradants for the drug substance. We look at impurities and degradants. For the drug product, we're really only required per ICH 6A, ICH Q3A and B are the USP, the EP guidances for impurities. And for drug products, you only need to look at degradants. You don't have to look at the process impurities that are coming out of your drug substance. And we'll talk about that here in a minute. So these are some specific tests, and this comes from ICH Q6A. Drug substance, solid oral dosage forms, all of the different dosage forms are on here, and this is not intended to be what exactly you need to put on there. You need to go through and determine what the critical quality attributes are, whether these things are applicable. For an example, in an oral liquid, it says particle size distribution. If it's not a suspension, you obviously don't need to look at particle size distribution. Same holds for topicals. Sometimes you have a suspension in a topical, and that's where you would look at particle size distribution. So you really need to know your product and understand it to determine which specifications of the specified tests belong on your specification. ICH Q6A, we've talked about a lot of it already, but in the back, there's a lot of decision trees. And the decision trees are there to help you decide what should go on your specification, and then also what the limit should be. So as an example, there's a decision tree for impurity and degradation limits. There is a decision tree for particle size criteria, for polymorph monitoring or control, chiral strategy, whether you have a drug substance that has some chirality and you need to, whether or not you need to have that on your specification, microbial testing approach, and then dissolution and disintegration. So as I stated, this is just a really broad view of specifications, because probably each one of these could have their own webinar and talk about exactly how to set specifications for them and what those look like. And most importantly, organic impurities. This is really where specification issues pop up. This is where specifications typically get really complicated and confusing. So I'm going to walk through ICH Q3A and Q3B just a little bit today, and then we'll get into it in a lot more detail and talk about exactly how to set specifications based on these ICH chapters. So Q3A is for drug substance impurities. And in both of them, they have different thresholds. One is the reporting threshold. Any impurity above for the drug substance, 0.05% would have to be reported. And then there's identification threshold, which above that threshold, then you need to identify your impurity. And above the qualification threshold, you need to have some safety data. So that holds true for both the drug substance and the drug product. But as I stated before, in ICH Q3A, the requirement is that you list any impurity, any impurity that meets the criteria that it needs to be on the specification, but you need to consider all process impurities, which would be starting material or intermediates. You also need to consider any degradants. But for ICH Q3B, which is drug product, you really only have to look at degradants. Sometimes you can have process impurities that are also degradants, so you need to keep that in mind as well. So on both of these, there's a couple definitions. One is specified impurities. Specified impurities are the impurities that are named on your specification. And the opposite of that, then unspecified impurities are impurities that are not specifically called out on your specification. So then we have unidentified impurities, which are impurities that we don't have a name for. And a lot of times the unidentified impurities would be listed with a relative retention time. But we don't know what it is. And so you can have unidentified impurities that are still specified impurities and unidentified impurities that are unspecified impurities. It gets very complicated. In the end, your specification needs to list all of the impurities that should be on there, and we'll get into what those should be. And then also you need to have a criteria where you assess that there is no unspecified impurity above the identification threshold. And you also need to give a total impurities specification. Again, we're going to have a webinar specific to impurities in the coming weeks. So other things that go on your specification are residual solvents and elemental impurities. And the residual solvents are ICH Q3C, and you have to consider any solvent that could be used or produced in the manufacture, purification of your drug substance, excipients, or your drug product. So it's kind of a risk assessment where you say, I know that these, the set of 10 solvents were used in the manufacture of everything that went into this, and then do an assessment of whether or not you need to test for that. So typically drug or residual solvents are controlled in the drug substance and in the excipients. And so you would have on your raw material specifications and your drug substance specification, you would list out the solvents that you need to test for there, and then have a report that says, this is my justification for residual solvent testing, that takes all of that into account and shows your control strategy for residual solvents. Elemental impurities is similar, but a bit different. It is a risk-based control strategy where you do a risk assessment, and you have to look at all possible sources of elemental impurities. And elemental impurities are heavy metals like lead, mercury, arsenic. And so this is based on ICH Q3D, which I think is on there, but you can barely see it. And so when you do that risk assessment, you say, what impurities could be introduced from my raw materials, including my drug substance? What elemental impurities could be introduced during the process, and what could come from the packaging, right? You have to look at maybe there's other sources in your process or in your drug product where heavy metals could come into it. And so you just need to do a full risk assessment on that. The elements that you have to look at are specific to dosage form. So when you're looking at injectables or orals or topicals, there's different classes in ICH Q3D, class 1, 2, A, 2B, and 3, I think. And depending on the route of administration, depends on what elemental impurities you actually have to look at. And in the end, what your risk assessment needs to say is that your drug product would meet the permitted daily exposure limits for the elemental impurities. And in the event that you have something where you are above that, then you need to remediate. You need to do some sort of change to your process or your materials to reduce that down to a safe level. So drug product specifications, our slide is a little messed up here, sorry about that. So drug product specifications, it's part of the control strategy. We're going to kind of talk about how all of these dovetail each other. It's possible to put tests or have tests on your drug substance that support the quality of your drug product, and not have to test that particular parameter in your drug product as well. So if you have things like polymorphism, sometimes I think you could argue that particle size distribution wouldn't change during your process. If you have data to back it up, a lot of this comes down to what data do you have to support your control strategy. So your testing that happens in your drug substance, if you don't expect or you have data that says that that criteria doesn't change, then it's something that maybe you don't need to test in your drug product. The same holds true for in-process testing. I think things like pH or maybe even particle size distribution here would be a good example. Again, if you're testing that in your in-process test or in your bulk, and you don't expect it to change during the fill, then that's something that you could test during your in-process, during the in-process testing, and not have to test again for your drug product. It still needs to be reported and it still needs to be assessed against the specification, but you might not have to test it twice. And then looking at your drug product release versus stability testing, the only thing that needs to go on your stability specification is the anything that's stability indicating. There are a lot of things that we've seen specifications where we're testing residual solvents or elemental impurities on stability, which just doesn't make any sense because we wouldn't expect contaminants to grow. Maybe you have some leaching that could happen there, but typically you wouldn't expect those kind of things to grow on stability. So in that case, we would be looking really at just the assay impurities, maybe a pH or possibly particle size distribution if you show that there is some change in that over the shelf life. One other thing to look at too is telescoping specifications. You can have a single specification. Typically, a drug product is 90 to 110% of the labeled amount throughout its shelf life. And you could have a in-process specification that is 90 to 110, a drug product specification that is 90 to 110, and a stability spec that's 90 to 110. But what could happen is if you release at the bottom end of that specification with method variability or things like that, you could fall outside of that specification at your release. And same with stability. Sometimes we see a decrease in the drug substance on stability, and if you're at the bottom end of that specification, then you could easily fall out. So a lot of times we have telescoping specs. So maybe your in process is 97 to 103%, your drug product release is 95 to 105, and then the stability spec is 90 to 110%. And that just kind of protects you from failing during shelf life testing. Pharmacopoeial considerations. We've talked a little bit about the general chapters and a little bit about monographs. Per ICH, where appropriate, pharmacopoeial procedures should be utilized. And I think that where appropriate, and the consideration you need to have, the thing that you need to think about when you're using a compendial method is that a lot of times for a monograph, that monograph was developed based on potentially just one manufacturer's process. So when we when we look at monograph methods for API or drug product, we have to be very cautious, and we have to have a really critical eye to what is listed in the monograph. As an example, if you have a API, it has a certain manufacturing process, it's going to have a certain list of impurities. The starting material, the intermediates, maybe even the degradants could be different for that API than the API that you're purchasing that has a different process. So in that case, you need to look at those impurities, make sure those are the impurities that they're similar to what you have from your manufacturer that you've developed. If not, then you need to make a change. Where that falls apart is method specificity, that the USP method isn't specific for the impurities that you have. But a lot of times the issue that we have is is in the LOQ of those methods, and then we just don't get enough sensitivity for them. So I think typically we would take the USP method, either modify it slightly to work for us, or really go with an in-house method. When you do that, you need to provide a justification of why you're not using the USP method or the EP method, and then you need to do a comparison of your method against the USP method and kind of point out where the issues lie and why you couldn't use the USP method. And I think we're probably, maybe 10% of the time I would use the USP method. So, and that really is just for the impurities in the assay. For things like, we would use the general chapters for water content and those kind of things. Methodology. We are an analytical testing lab. So we take our methodology pretty seriously. We'll have separate method validation and method development talks as well. But method considerations, when we're talking about specifications, seems kind of obvious, but isn't necessarily. But your method needs to be accurate and precise at your specification limits. So if you have a drug substance method and that specification is really tight, you need to make sure that your method is also really precise. You can't have a method that allows like 3 or 5% variability when your specification limit for that drug substance is 98 to 102. So when you're developing your methods, you might not always be able to use the same method for your drug substance and your drug product. You need to take that into account. You need to make sure that your method is specific for all of the analytes. All of the analytes are even the impurities that are not listed on your specification. You need to be specific for those with the ones that are listed on your specification. You need to make sure that your drug product method is specific for all of the excipients that might exist in there. And then you need to make sure that your method is stability indicating. And what that means is that you've done some forced degradation work. You've looked at the degradation products that are impurities that are possible. And the method, those degradation products are specific to each other, and then also specific from the API. If your API peak has a whole bunch of degradants under it, you're not going to see that variability, or you're not going to see any degradation that's happening. One of the big things too that I think gets overlooked a lot in methodology and specifications is having your method consistent from early development all the way through commercial launch as much as you possibly can. It kind of is like a cart before the horse situation of how much method development do you do in early phase, knowing that this product or this project could be canceled. How much time do you have to put into it? But if you can do the work at the beginning to characterize your impurities, to do some rough force degradation, a little bit of R&D stability, excipient compatibility, all of those kinds of things early will get you a method that's specific, that then you can kind of read that data across as you get to the end of development and you're going commercial and you're doing your filings, you're justifying your specifications. If you didn't know about this one impurity, or you didn't have specificity of something, then you've got gaps in your data that then It just doesn't give you a very good body of data at the end. So doing the work at the beginning really helps you as you get to the end and justifying your specifications. Acceptance criteria. So this is a really big one. Your acceptance criteria needs to be set based on, first and foremost, data that you have. So the tox lots, your clinical lots, all that batch data. Let's say you've got 5 batches that you've made. They're all, from, pilot scale up to, probably don't have any commercial scale when you're setting your acceptance criteria. But looking at all of the data that you have, compiling it all together, looking at the variability in the, from lot to lot variability for things like impurities, things like for topicals and whatnot, pH or viscosity, that's where you kind of really get, really need to look at those. I don't think we put enough time into those specifications sometimes. Looking at your development data, that excipient compatibility, R&D stability, force degradation, take into account analytical variability. We talked about that really tight drug substance specification. If you're allowing 3% variability, it's 98 to 102. If you're releasing at 99, there's a good chance with analytical variability, you're going to fall outside of that. So keep that in mind too when you're setting your specifications. Manufacturing variability, your stability behavior. Do you see any trends on stability? Looking at your telescoping, saying, okay, well, we need to set our release spec on our drug substance really low for this one impurity because it's also a degradant. And we see it growing 5-fold on stability. So it needs to start really low. We can't bring something in that's really high. Looking at any ICH or USP limits that exist. And then raw material and drug substance data. An example that I have was for a topical that we were that we're doing the justification of specifications on. We would have meetings where we'd get together and we'd have to justify our specs. And in that meeting, we were trying to argue that we really had limited raw material lots that went into our product. And therefore, we weren't really sure on our viscosity if we were going to see some variability there. We just didn't have the data. The answer to that is generate the data, but we didn't have it and we needed to go. So we were arguing for a wider spec because we just really didn't know. Ended up going with a tighter spec, and sure enough, first clinical lots had issues on meeting that viscosity specification. So that's something that you want to take into account. I think that we say that hindsight is 20/20, and I think that is really important with acceptance criteria. A lot of times everything looks great. There's no reason why we're not going to meet this specification until we make that lot and we don't meet that specification. And I think anybody that has gone through product development has lived that pain. And then you learn and you say, I need more lots of this viscosity, of this raw material that supports the viscosity of the product. I need more lots of that raw material in-house so we can look and see what the effect is there. Reduced testing. So reduced testing, there's a couple of them. One is periodic and skip testing, and this is in the ICHQ 6A, so you can refer to it there. But periodic or skip testing, and then parametric release. So periodic or skip testing is when you don't test every single lot for this one parameter. This is used a lot in raw material testing. We would test the first 3 lots, kind of qualify that raw material with that supplier's material, and then only have to test 1 lot annually. You need to have the frequency of the testing indicated on your specification. You need to have this criteria or this concept, right, approved by the regulatory body. And then if you fail to meet any of the criteria on your specification, then you have to report it, and then you probably will have to go back to full testing until you can figure out what happened and get back into meeting those criteria. Parametric release is where you're taking some operational controls that you have. A lot of times this is around sterility. So it's an alternative to release testing, and it has to be validated and then revalidated. Typically, you would revalidate annually, and this also requires pre-approval. Justification of specifications. The way I typically handle this is with kind of a massive report that starts with the drug substance specification and talks about where the criteria came from so the different tests, why the tests that are on there, justifies the exclusion of any tests, things like polymorphism or particle size. If your API is dissolved and you don't expect there to be any issues during processing based on the particle size, you don't need to do particle size testing. And then goes into the in-process, the release, and stability specifications. We don't typically put raw material excipient specs in that justification. We would put that just in the specific CMC section for those. But the big thing on your justification of specifications is it needs to be based on batch data. So this is where you put those 5 lots that we talked about. Maybe 3 were at small scale, 2 of them were scale-up batches. You have your drug substance data in there. What kind of impurity profiles are you seeing? Hopefully you've got 3 lots of drug substance that you can put the data in there for. Any stability graphs you have, anything that's trending, putting that information. And then a summary. This one's really important, is the summary of the impurities and the degradants. You need to list out every possible impurity that could be in your product, and then provide a justification of whether or not that could be in, whether or not you're going to see it. Is it a process impurity? Is it a starting material? What is the structure of it? and then indicating kind of your control strategy for that. And then you want to talk about your kind of throughout is your control strategy for whether you're telescoping your specifications, or if you're choosing to test something in your in process testing and not testing it at release, then you need to talk about that. And then any kind of reduced testing or parametric release, you need to talk about that there. There is, to me, a newer ICH guidance, and then ICH Q12, which is talks about product life cycle. So I was kind of looking into this in preparation for this webinar, and there was a webinar by the FDA last year, and it looks like there's about 16 people that have provided information about their established conditions. And the whole thing about ICH Q12 is reducing down your post-approval filings, your CBE 30s or CBE 0s, and kind of setting up, they talk a lot about established conditions. Specifications are established conditions. And if you make a change to it, depending on how much data you provide to them, it could be an annual reportable versus a prior approval supplement. So product life cycle, FDA, other regulatory bodies really expect you to learn more about your product. And as you learn more, make adjustments to your specification. So adding or removing tests as your control strategy matures. And this is from not just like post-approval, but also during development. So kind of one of the questions is how much do we have to have on our specification when we're like, phase 1, phase 2? And the answer to that is put what you know on there. And in some cases, just put report, because you're generating that data. There's certain areas where you would need to have more control if there's a safety issue or concern. Certainly you want to have a specification around your assay, but maybe it's just a little bit wider so that you can, you're sending stuff to the clinic. Is it going to be 90 to 110 throughout the whole clinical study? If it is, then you can have a lot wider spec. So that you can add or remove tests, you can tighten or loosen your specifications as appropriate. If you learn that with a pH, when pH gets, we're at the top end of our specification, we're actually seeing more degradation of our impurities, so we need to tighten that pH spec, then that's what you're going to do. And that's really what the expectation is based on ICH Q12. So building the specification, again, really broad strokes, because this is dose route of administration, so dosage form dependent. It is API dependent. It is just just so many things go into building your specification, but this is a process that I go through in building a spec. I gather information first. So I would make a Word document that lists out, what is the active ingredient, what are the strengths that we're looking at, the excipients, is there a USP monograph or an EP monograph? What kind of information do I have on this? And then kind of put all of that together, then go in, you're doing your development work where you're characterizing the product, you are looking at degradants, excipient compatibility, you're looking at viscosity, hopefully with a whole bunch of different raw material lots, and then you determine your control strategy. So I'm going to test, I know that this doesn't change from in process to packaging, or this is definitely not a stability indicating parameter. So trying to determine early on, and a lot of this stuff can be done pretty early. Then you go into designing your methods based on all of the information you have. Hopefully now you know what impurities you're looking at for your impurity methods, which of those impurities are degradants, what kind of levels you're seeing for all of those, what the excipients that you have, is there any variability that's going to affect the specificity of my methods? All of that. How tight does my precision need to be able to meet these specifications that I'm going to propose? And then assess the data, set those acceptance criteria, go back, because it can be really iterative, do some more characterization, maybe modify the method a little bit before you go into validation. But so then once you have all of that, you set your acceptance criteria. If you're in early phase, you come back in as you're moving through the phases of development and reassess your specification. Look at the lots that you're manufacturing. Are you seeing anything new? Is anything different? Do we need to change anything? Is there a new impurity that's showing up on actual accelerated, stability rather than the R&D work that you did? So some common specification pitfalls. The biggest one is it's not based on CQAs. And that's a critical quality attributes. So you need to look, is this a critical quality attribute of my product? If it's not, don't put it on there. If it is, and you don't have it on there, then that could be a problem. You're not adequately justifying your specifications. You're setting a spec based on limited data or what you've seen someplace else. Your limits are too tight. That's a really big one, because then you're getting out of specification results. You're fighting that all the time. Unnecessary tests, we already said. No impurity strategy. This, honestly, if there was one pitfall on specifications, it is impurities. And whether or not you have done the work to understand what truly are your impurities in your product, rather than just kind of blindly looking at maybe somebody else's product, or really not taking a kind of a holistic assessment of your product. And then limits tighter than the method capability. I think that one is a big one as well. So to summarize, specifications are one part of a total control strategy, and they're designed to ensure that your product quality and consistency are there. Each test criterion must be justified using development data, clinical and tox lot data, stability data, and then considering the expected variability, not just from your process, but also from your raw materials and your analytical methods. You want to have specifications that confirm your quality, but they don't need to establish full characterization. If it doesn't need to be on there, if it's not a critical quality attribute, it should not be on your specification. Acceptance criteria should be established, and again, reflect the expected analytical variability and your method capability. And then where reduced testing is proposed, then the approach must be validated and scientifically justified. All right, and then that is it. So we've got a little bit of time left for questions here. I'm going to look and see. All right, so one question that we had is, what's the fastest way that you can spot a specification that's going to give issues with your operation? and I think I kind of said this a couple of times, specification or impurities. When you look at the impurities, if they're not based on the ICH requirements for the ID or qualification, you're setting your impurity specifications too tight. Maybe they could be justified to be wider. You have impurities that are consistently on stability, getting up to maybe 0.3% label claim, and your specification is 0.3%. That's what really is the specifications. That, or looking at it and seeing something like polymorphism in a drug product where it really isn't a critical quality attribute. What do you need? So can you use a different method than the monograph, and what you need to show if you do use a different method? You can use a different method than the monograph, and you need to justify it and say this is a reason why. We would always put a statement that said that in the event of a dispute, the USP method, the compendial method prevails, but then would say that the method's not specific for all of the impurities that we're looking at, and that was a pretty strong argument to not using it. But you also need to do a comparison. You want to say it's equal to or better than the compendial method. You certainly don't want to swap out a HPLC method with a titration method, because the titration methods are really non-specific. So if you have to do that, you better have a pretty good justification for it. If the monograph doesn't fit your product, like matrix or strength or route, what is the cleanest path forward? I think that's the cleanest path forward is to take the monograph method. In the end, then that monograph method is not suitable for use. It's not appropriate. So let's see. And so then I think that's a really good justification for not using it. The next one, how do you set limits when you have very little data? You generate more data. That's not always the answer. A lot of times we would use some statistical analysis if we're looking at stability and we are not out to 24 months yet, which is the proposed shelf life, then we would trend the data. And based on that, have a proposed shelf life, whether or not we would hit the 24-month or not. Then I think if you can generate, maybe you can generate some lab scale data or use other statistical. Sometimes it's like, 3 standard deviations or looking at confidence intervals and stuff like that. But I think a lot of times it's better, especially if you're doing a filing, to go in with a little bit wider specs, because hopefully over the course of the 6 months to a year it's going to take for you to get your product. I'm being certainly not a generic timeline, but 6 months to a year to get it approved. You're generating more data, and before you have approval, you'll have enough to really hone those specifications. When do you need a full validation for a method versus a phase appropriate qualification? Once you hit phase 3, you need to have full method validation. Before that, it can just be a qualification. The differences between a validated method and a qualified method are things like robustness and force degradation to show the stability indicating aspects of it. But a lot of the robustness and force deg really should be done in the early stages in your method development. So you should already know a lot of that, but it is a less cumbersome of a protocol if you're just doing a qualification. Sometimes you don't even need you can just do a report. For residual solvents, which solvents are reasonably expected to be present? That is typically determined by a residual solvent statement that you get from the manufacturer of your excipients. If they can't give you a residual solvent statement, you should go find another supplier, because that's a really good indication that they're not really pharma grade. So if you get the residual solvent statement for every single one of your excipients and look at those, that'll tell you what reasonably can be expected to be present. You need to take into account too, not just the solvents that are listed, but then potentially any impurities. I know benzene is an impurity of some solvents, so in the event that, so you need to keep that in mind too. And then anything that you're using in your process, if you have a solvent in your process, you should be looking at that. What are top mistakes when drafting the first specs? I think probably hammered this home pretty hard. I'm going to go back to description and color and appearance. It seems so simple. pH viscosity specifications, it seems so simple, and those really are the things that trip you up. I think also impurities can it probably equally as difficult. So I think that that's really where it is. There's so much to keep in mind. You're really, when you're setting specifications, are looking at every single aspect of your product. A lot of times your process, you have to look at your formulation data, your development data, all of that kind of stuff, and take all of that into account. And just like method development, it's kind of like a learn as you go kind of thing. And the last one I had was, what is the most common reason specs get challenged during a review or inspection? I think impurities, dissolution probably as well. I think those two are not having an adequate justification for what you're doing. I think that really is that's what I've seen anyway on reviews. Also, there's it just seems like we just have new things that particularly the FDA is looking at, and so whatever is the problem of choice. We just have another one. What phase stage do you need regulatory approval to change the product specification versus just informing? I think that that's a really good question. I think you just need to inform. If you've got an IND that you filed, you need to inform as you're modifying that specification. Once you have approval, it really depends on the agreed upon control strategy that you have with the FDA. If you're going the ICH Q12 route, then you need to have, but typically you would need to get approval. Unless you have prior approval to just do an annual reportable, you're going to need to get approval prior to making those changes. So excellent. And that is all we've got today. This is our first in the series. We'll be coming up here with another webinar for, I think the next one is going to be analytical development, which I'm really excited about because it is what I love and what I do. If you have any questions, you can email us at engage@triveritylabs.com. We would love to work with you with your pharmaceutical product development. We would love to help you set your specs. If you can't tell, I get super nerdy and pretty excited about specification setting because I think that it's kind of the icing on the cake of all of the analytical work that you do kind of gets culminated into these specifications. And when it's really well done, it's good for everyone. So thank you all so much for joining us, and we'll see you next time.